What's up everybody, Organized Biology here, and today we are kicking off our membrane-bound organelles series with the nucleus. Probably one of the most important organelles in your cell. So brief review, what is an organelle? An organelle is any small little organ bound by a membrane within your cell that'll have a specific function for the cell, okay? So today we're going to talk about the most recognizable one. If you look at any cell, a lot of the times you'll see a big dark dot in the center. That big dark dot is the nucleus. And I've actually blown up the nucleus here so we can see specific structures inside as we go along in this lesson. So let's start. What are the functions of the nucleus? Well, number one, the main function of the nucleus is to store DNA. Now, you've probably heard of DNA before. It stands for deoxyribonucleic acid. I want you to remember these as the instruction manuals for your body's cells, okay? So in order for the cell to know what it is and what it does, it needs to use the instructions in DNA. Now remember this, I'm going to talk about it later. All of your cells have the same instructions, and that's going to be interesting, okay? Because they all look so differently, how could they have the same instructions? We'll talk about it in a second. Now, what is the DNA? Well, DNA is a double helical structure, okay, made of base units called nucleotides. And each nucleotide is going to contain a nitrogen base that is either going to be A, T, G, or C. Now, if we look at DNA, here's your double helix. Okay, the backbone is made of a phosphate and a sugar, and then the nitrogen bases are all the letters. As you can see, there are 4 letters, and I'm going to write them out. A, T, G, and C, and they're always going to bond in that way across from each other. That's what it means to be double-stranded. You got 2 strands on each side. And all of these letters, think of them as basically the code, okay? So depending on how the letters are organized in a row, it's going to code for different amino acids. Amino acids are very, very important. Think of these as little Lego blocks that are going to join together to form some sort of beneficial structure called a protein. So many amino acids chained together are going to be turned into proteins, which gets to my second point about the nucleus. The nucleus has to regulate which DNA is actually read into these proteins via making amino acids, because the proteins themselves are the function and structures of all cells. So if you have a question like, what determines what a cell is? It's what it's made of, what proteins it's made of, okay? So remember that. Now, here's a key issue then, right? If all of our cells have the same DNA, but our cells have different proteins, well, how do the cells actually know which proteins to make in order to function properly as that cell? So for example, how does a skin cell know how to make skin cell proteins like melanin and keratin? How does your muscle cell know how to make proteins like myosin and actin? Here's the key. The nucleus has to organize itself in such a way that the proper DNA instructions are read in different cells. Okay, so for example, if this is a skin cell, for example, we are going to place different segments of DNA along the outside, and we're going to loosen them up a bit. And each of these segments of DNA are going to be important for the structure and function of the cell. So for example, let's say this little DNA segment codes for the protein keratin, and this one codes for melanin. 2 very important proteins for your skin cells. They are going to be found on the outside where it's easily accessible. Now I'm going to get to that here in a second. But this region of loosely bound DNA on the outside, so this whole region is called euchromatin. Now U refers to true. So true and chromatin refers to chromosomes, chromatin, you may have heard of these. This just refers to any sort of DNA segment, okay? So it's the true DNA of that cell, and it's the DNA that's actually going to be read. And I'm going to get to that here in a second. Now, if there's true DNA called euchromatin, there's also maybe false DNA in a way, or other DNA, and that's what this region in here is called. This whole region of really dark DNA, really condensed DNA, is called heterochromatin, meaning other chromatin that will not be read. So things in this center of the nucleus are not going to be read. Therefore, these DNA segments that code for different proteins that don't pertain to skin cells will not be read by that cell. Really interesting. It's the job of the nucleus to organize itself in that way. Now, getting back to easily accessible, right? Look at how loose the DNA is on this outside euchromatin area versus here. Here, it's going to be very easily accessed by a little machinery enzyme called RNA polymerase, which I've designated by these little blue squares. So this word itself means it's an enzyme, ACE, basically something that does some sort of chemical reaction. Polymer means some sort of long chain of RNA, okay? So it's basically little machinery that's going to produce RNA. Now, what is RNA? I want you to think of RNA as a recipe, a transcribed recipe of the DNA, okay? So unlike DNA, it's going to be single-stranded. So if you look at RNA, it will look something like this. It'll be a single strand of nucleotides, replacing the T with U, interestingly. You can watch my protein synthesis video for more details on this in a separate video. I will link it here. But this is a single-stranded recipe that we basically transcribed from the DNA, and now we need to send it out of the DNA to be produced into amino acids, thus proteins. So here's a kicker. Why is RNA recipe single-stranded? Because it can actually fit through these tiny little holes called nuclear pores. Okay, so point at these are called nuclear pores. So the DNA can't leave because it's double-stranded, it's too big, but the RNA is single-stranded, so it can actually leave the nucleus. Now, here's a problem. We have the recipe, right? We've got the little AUG that I wrote here, but the problem is we have nobody to translate what the heck that means. And in order to translate it, we need ribosomes, okay? You've probably heard of ribosomes as little protein-making factories, but I don't have one right now, because the nucleus also produces ribosomes in a region called the nucleolus. That's this blue dot right here. This is called the nucleolus. Very small change of words. I hate when biologists do that, right? Now the nucleolus is going to produce what's called ribosomal RNA, which will form ribosomes that look something like this, okay? So that nucleolus is going to produce the ribosomal RNA, thus producing ribosomes. And I want you to think of these as little protein chefs. Ribosomes are protein chefs. So what they will do, and I'm going to skim through this real quick, is they're going to take that RNA, they're going to read it, and they're going to spit out the proper amino acid. So maybe the amino acid is going to look like a little triangle, looks like this. And as it reads the RNA, because in reality the RNA would be pretty darn long, it will continue coding for different amino acids, different shaped amino acids, eventually producing the protein. Okay, and that protein is going to give that cell its structure and function. Now, this is called translation. Okay, we are translating the mRNA transcript into a protein, whereas in this first one, we were actually transcribing it. So this first step was called transcription. Okay, so there you have it, transcription, translation. So here's your DNA in the nucleus being read for the proper protein for that proper cell to do its proper function, okay? So in summary, the nucleus stores the DNA, regulates which DNA is read into the proteins based on the structural components within the nucleus itself. And we also have the nucleolus producing rRNA, ribosomal RNA, to help produce the ribosomes. We also will use RNA polymerase to produce mRNA recipes to go to the ribosomes to make the proteins to give the cells its structure and functions. This has been Organized Biology. Thank you so much for watching. If this is helpful, please like the video, subscribe to it, and comment if you have any questions. Thanks again.